DNA replication. DNA is made by a bunch of these nitrogenous bases with a nucleotide. Nucleotides are made up of an oxygen and are held together by a phosphate group and a sugar, deoxyribose. That is only in DNA. Then for it to make a double helix for DNA, a structure, each nitrogenous base needs to connect with its pair. Each pair is held by hydrogen bonds. A goes with T, or adenine as it's called, goes with thymine, and it's held together by a 2 hydrogen bond. Cytosine and guanine go together, and they're held together by a slightly stronger bond made of 3 hydrogens. Each of these are going in opposite directions. This would be the leading strand, would go from 5 prime to 3 prime, which is just the phosphate group is on the fifth carbon bond of the sugar, and on the next one it's held on the third carbon bond. This side goes in the opposite direction from a 3 prime to a 5 prime. You kind of tell by the direction of where the oxygen is pointing. Is how you can tell. So now let's just get to the separation. Okay, so first off, we start with the helicase unzipping. Helicase will just come in, kind of tear these things apart, just take out the glue that holds them together. This part is called the replication fork. There's going to be 2 strands, a leading strand and a lagging strand. RNA primase goes in through the leading strand from one end and sets up a sort of base where the DNA polymerase can come and start off in. DNA polymerase starts off at the RNA primase base, goes in and fills in the rest of these nucleotides and gives them their opposite connecting bases. Helicase still holds that open. It'll be there the whole time to keep this whole chain open. DNA primase comes in again, or RNA primase comes in again, sets up another little starting off point for DNA polymerase on the lagging strand. But since this leading strand goes from 5 prime to 3 prime, RNA primase only has to set it up at the beginning so that DNA polymerase can just come in and finish it off. But since this lagging strand goes from a 3 prime to a 5 prime, RNA primase has to come and set up a starting point somewhere in the middle because it can't really go against it. So it just sets it up in the middle, goes down, and then sets up another one here at the end, like this. And then DNA primase comes in and fills up that row. And then whenever the DNA primase comes in and fills up the rows, they're called Okazaki fragments. There's a second version of DNA primase that comes in, makes sure that all of these RNA primase bonds that were made get turned into actual DNA so that they actually connect. And then DNA ligase comes in the lagging strand, and it kind of just like shuts those bonds together so that they actually hold.